Hi, I'm Dr. Wilson. I'm a PhD molecular biologist, and welcome to another COVID debunking video. This week, I'm going to be covering a paper written by anti-vaxxer Peter McCullough, who was on the Joe Rogan podcast and spread a bunch of disinformation there, and his fellow anti-vaxxer Stephanie Seneff. This paper is clearly not written for scientists. It's written for the general public and for journalists. Because when you actually read it, it's just a mess of disparate claims and descriptions of biological mechanisms, but they never actually link any of those mechanisms to COVID vaccination with any hard biological experimental data. It's a generous take to call it speculation, but at best that's what it is. It's nothing concrete, but they frame it as demonstrating a mechanistic link to COVID vaccines and all of these adverse events that they talk about. Papers like this that are written for journalists and the general public and not scientists are always a huge red flag, but they always make a big splash in anti-vaccine circles. So here I'm going to break down and summarize some main reasons and examples as to why this paper is just terrible and shouldn't be taken seriously by anybody and probably should never have even been published because scientifically it's pretty useless. Let's go to this example of this point in the paper where they talk about G4 quadruplexes in the mRNA vaccine construct. Now, G4 quadruplexes, I think they chose this topic because it's something that journalists and the general public probably won't know much about, but they can spin it in a flashy way that makes it sound really important and really scary. So what are G4 quadruplexes? Well, they are structures that DNA or RNA can take. It happens when there's a string of Gs or guanines in the sequence. These nucleotides can undergo what we call Hoogsteen base pairing and stack on top of each other in a planar manner to create a secondary structure that looks kind of like this. These structures can have regulatory impacts on the expression of a gene. There are several different structures and features in a gene that can influence its gene expression. For example, there are features that can cause a gene to be transcribed less. This means that the gene makes less mRNA and therefore less protein. This can go the other way. There are features that can enhance the amount of mRNA it makes. There are also features that can function at the stage of translation. So once the mRNA is made and a protein then has to be made from that mRNA, that can also be controlled as well. G4 quadruplexes fit in this category of features of genes that can influence gene expression depending on the context and which gene it is. McCullough and Seneff talk about this in the context of cancer, and it is true that G4 quadruplexes have been found as regulatory mechanisms in certain genes that play particular roles in cancers. But where this section and so many other sections of this paper fall completely flat is when they try to link these biological mechanisms to the COVID mRNA vaccines and say that it's going to cause cancer. They're really grasping at straws here. Just because one regulatory mechanism plays roles in certain genes that contribute to cancer progression or suppression, that doesn't mean that those same regulatory mechanisms in a completely different construct delivered in a vaccine is going to have any effect whatsoever on cancer. The best way I can describe this gigantic reach for a straw by McCullough and Seneff is if they were saying that turning on your bedroom light with a switch will also affect your oven downstairs because both use electricity to be switched on and off. That's pretty much what they're saying here. G4 quadruplexes are just a regulatory mechanism. Just because they function in cancer genes doesn't mean that if they're found to help regulate the mRNA on the COVID mRNA vaccine construct, that's going to affect cancer somehow. They're completely separate. In order to demonstrate their claims, they would have to first show that G quadruplexes in the mRNA COVID vaccine constructs play some functional role and exist. And then they have to demonstrate that it's those structures particularly that are somehow interacting with cancer genes and contributing to some sort of pathogenesis. But they don't even do that first step. The furthest they go is referencing a very lazy paper, which just suggests that G4 quadruplexes might form in COVID mRNA vaccines. They don't even demonstrate that that is true, and it would take a really simple experiment. It's just called circular dichroism. It's a method that can easily identify whether or not these G4 quadruplexes have formed in a nucleic acid sequence. They don't even do that. It's the bare minimum laziest thing that they could do to just scare their readers and have journalists report on their paper as if it's meaningful when really it's not. And that's really the pattern that this entire paper follows. They describe biological mechanisms, sometimes correctly, sometimes incorrectly, and then fail to link those mechanisms in any meaningful clinical way to COVID mRNA vaccines. Usually, what they do is they reference in vitro or cell culture studies. This is where there are cells grown in a dish and they are treated in some way. But these experiments are usually a start to a hypothesis. Usually, you have to do the experiment in cell culture, get a clue as to what is going on, and then verify it in further experiments. And in this case, that second part is really, really important because cells in a dish are going to behave very differently from cells in your body. But really, none of their sources actually do that. None of them actually link the COVID vaccine to these pathological mechanisms that they describe. In fact, a lot of the time, they actually describe these pathological mechanisms occurring in SARS-CoV-2 infection. Yeah, they will describe how a SARS-CoV-2 infection causes these problems and maybe how it interacts with some of the biological mechanisms that they're describing, but then they just assume that that same thing happens in vaccination. But vaccination, as we know, is very different from an actual infection. For example, McCullough and Seneff spend a lot of time in this paper talking about interferons, which is a part of the innate immune response that helps the body respond to viral infections. They explain that patients with COVID infection who had a poor interferon response were much more likely to have severe COVID. But then they don't really link that to COVID vaccines in any meaningful way. They claim that COVID vaccination doesn't produce a great interferon response and that it only focuses on memory cells. That's somehow a bad thing? That's what you want vaccines to do. You want vaccines to elicit an immune response that instills a really good memory in your cells so that when you encounter the real thing, you can respond to it really well. And that's exactly what we see. There's no evidence that people who have been vaccinated have a poorer innate immune response when it comes to other viruses or to an actual SARS-CoV-2 infection. They do reference one strange paper that looks at 11 patients who received an inactivated coronavirus vaccine, so not an mRNA vaccine, and they look at their immune markers starting from day 0 when they got the shot to about 90 days later. Along their timeline, they found some changes and some differences in some parameters, but this is never connected to any functional clinical significance or whether or not this is just random variation within a population. And with only 11 patients, that is far from ruled out. When we look at real-world data, we see that COVID vaccines are in fact protecting people against severe outcomes of COVID-19. But despite this, they still try to scare their readers by talking about some of the more recent anti-vaccine talking points that have emerged in the past few months. A couple of months ago, a paper was published in the journal Cell, which showed that spike antigen following vaccination can be found in the germinal centers, which are structures in immune organs called lymph nodes of vaccinated individuals for several weeks after vaccination. The authors explained in detail how this observation explains the very robust immune response that vaccines can elicit. What happens is when the spike protein, which is the antigen in this case, is seen by the immune system and processed by the immune cells, it is taken to these germinal centers in lymph nodes. It is here where these inactive spike protein peptide fragments are presented by immune cells called antigen-presenting cells. They present this antigen to B cells, which will constantly learn from that presented antigen and use that information to create better and better antibodies. It's like a school for B cells, so to speak. Previous literature has shown similar phenomena with some flu vaccines, where persistent presence of antigen in germinal centers correlated with a more robust and longer-lasting immune response. So to quickly sum all that up, the spike protein is gone relatively quickly from most of the body except these immune organs where they're supposed to be after vaccination. And that presence in those lymph nodes helps create a more robust and better immune response over time. Here, McCullough and Seneff actually bring up the by now very old claim that the spike protein can inhibit a process called VDJ recombination, which is a form of DNA breakage and repair that your immune cells naturally do. This process normally happens in the bone marrow in immune precursor cells. It happens so that they can expand their ability to react to theoretically almost any antigen that they could ever recognize. It's a fascinating process, and I'll link in the description some papers if you want to read more about that. But all you need to know here is that it happens in the bone marrow, and it is important for immune function. So in order for the spike protein to disrupt this process, it would have to reach those immune precursor cells in the bone marrow, make it into the nucleus, and then disrupt the process. So does that happen? Absolutely not. McCullough and Seneff reference this paper, which claims to show that if you overexpress spike protein in really high amounts in cells that are cultured in a dish, then you can drive it into the nucleus and disrupt this process. They also show that several other proteins in the SARS-CoV-2 genome do something similar. The big problem here is that this is done by getting cells to continuously express a high amount of spike protein in a dish. This is not physiologically relevant when it comes to vaccination. This is not what happens, and it's not what happens to the bone marrow cells that we talked about earlier. In order for these findings to have any bearing in reality, they need to be shown to occur in an animal model or humans, and that's not been shown. In fact, the exact opposite has been found. We see that the vaccines do elicit a very robust immune response, which would require VDJ recombination to have occurred previously. And by the third dose of the COVID vaccine, we see that the repertoire of antibodies that are produced by this immune response expands significantly. This would not happen if VDJ recombination was inhibited. This paper was really bad in its interpretation, and now Peter McCullough and Stephanie Seneff are referencing it, so naturally their interpretation is just as bad. If you want to know more about this paper, it was actually reviewed by the folks over at This Week in Virology. These are experts in virology and immunology. They reviewed the paper in detail, so if you want more about it, you can go check them out. Outside of all of the in vitro stuff that they wildly misinterpret, they turn to, of course, VAERS, the Vaccine Adverse Event Reporting System. And like most anti-vaxxers, they use it incorrectly. Essentially what they do here is they look at VAERS to see increases in reports of diseases that are consistent with some of the mechanisms that they explained earlier. For example, they look at reports of Alzheimer's and cancer, and even though these diseases take years to develop, they claim that somehow this is evidence that mRNA vaccines are causing these things. In reality, it's just the fact that because these vaccines were emergency use authorized, there is a bigger influx of reports to VAERS. And also the fact that VAERS is self-reporting and anybody can submit any report that they want without it being medically verified. And the fact that hundreds of millions of doses of these vaccines are being given in a very short span of time in countries like the US that have systems like VAERS. But no, none of that comes up in this paper. They simply take the reports at face value and say that it's evidence of their mechanistic speculation. No, that's too kind of a word. Mechanistic fantasies, let's say. The correct way to use VAERS would be to follow up on those reports, clinically verify them with medical records or any other kind of patient follow-up, and then compare those confirmed rates of cases to the background rate that you would expect in a normal year. There are several studies that do that, and they find consistently that COVID vaccines are extremely safe, not associated with things like cancer, Alzheimer's, or other serious outcomes. They are associated with myocarditis, but I've already made a video on that and how COVID infection is much, much more of a risk than taking a vaccine. So instead of talking about what they say about myocarditis in this paper, I'll just refer you to that previous video that I made. But of course, none of these papers that do this proper analysis of VAERS data are referenced in this paper, and they don't try to do any of that analysis themselves. Again, it's all just taking reports at face value, which is not the correct way to use VAERS. That all pretty much sums up the quality of analysis that you'll find in this paper. It doesn't add anything to the field scientifically. It makes wild speculations, and again, I use that speculation term politely because speculation at least has some evidence behind it, but this paper doesn't manage to do that. It doesn't manage to support any of their claims with any solid evidence. Instead, they describe mechanisms and then say that they think that this non-evidence supports their ideas, and then they challenge the scientific community to follow up on their ideas and actually test them. And that's just not how a useful paper works. That's not how scientists write papers and communicate with each other in the literature. Again, this paper is written for the general public and for journalists. It's not written for scientists because if any scientist that knows what they're talking about reads it, they'll see that it's useless. It doesn't cite the pertinent literature that describes the immense safety and efficacy of vaccines. Instead, it makes these tired anti-vaccine claims, saying that antibodies induced by COVID vaccines fade in 3 to 10 weeks after the second dose, which we expect. We expect the immune system to contract and for you to be left with immune memory. And that's what we see. They say that people are being advised to seek booster shots at regular intervals, and this is also misleading. 2 to 3 doses of COVID vaccines are going to be good for the vast majority of people. It's only the most vulnerable people who should consider getting a 4th booster at this point in order to prevent themselves from infection in, say, the coming fall, where we would expect another surge of COVID cases that might threaten the most vulnerable who don't stand to benefit from the vaccine as much as others do. They say that emerging variants such as Delta and Omicron show resistance to antibodies induced by vaccines. And even though this is true for Omicron, that it is good at evading antibodies, it's not so good at evading the rest of the immune system that can prevent disease. As we see, COVID vaccination is still really good at preventing severe disease from Omicron. And finally, they say that it has become clear that vaccines do not prevent transmission of the disease, but can only be claimed to reduce symptom severity. This is extremely misleading. As I've explained several times on this channel, the data clearly show that COVID vaccines reduce transmission. They don't prevent it entirely, but that's not the point. The point is to prevent disease. The point is to prevent people from getting into the hospital and into the morgue. It's to keep people healthy and alive, and they accomplish that really, really well. But none of that is highlighted at all in this paper. Like I said before, this paper offers nothing useful to the scientific literature. It only serves to fuel anti-vaxxers. That's it. Actual scientists are meticulous and careful about their ideas. They want to see the data. They want to do their own experiments in order to test their ideas before they publish it to the world and have the scientific community critique it, because they care about their colleagues' impressions of them. They care about the quality of their work. But if you're not trying to appeal to other scientists and your colleagues, you're just trying to appeal to laymen and journalists, then this is what you get. But bad papers do sometimes get published, and this one did. So it's going to be fueling anti-vaxxers, and as long as it's doing that, you can arm yourself with the knowledge that I explained in this video and all of the links that I've provided in the description, so that you can at least know that this paper is garbage. Well, that's going to do it for this week's video. Again, like I just said, all of the links to all of the science that I talk about in this video are going to be in the description below so that you can read them for yourself. Next week, I'll be getting back to my review of RFK Jr.'s terrible new book about Fauci, and I'll be getting into Chapter 5, which is also, unfortunately, about HIV/AIDS. And man, it's really bad. If you thought I was mad about Chapter 4, I'm also going to be mad about Chapter 5. So as always, thank you for watching. I do hope you've enjoyed it, and if you did, then don't forget to subscribe so that you can catch me next time in 1 or 2 weeks where I'll be debunking some more funky stuff in RFK Jr.'s book. See you then.